Today we are going to see some of the experiments done by the famous scientists. Let us see first one. Griffith's experiment on bacterial transformation. Pneumonia is caused by one of the bacteria called Streptococcus pneumoniae. This bacteria is having two strains. The first is called smooth virulent strain, written as S3. Other one is rough avirulent strain, written as R2. Let us see why it is virulent. Virulent means which can cause disease. Polysaccharide capsule was seen outside this bacteria. The red color is a capsule which was smooth and shiny. WBCs cannot engulf such type of bacteria inside the body of an organism. So this S3 cannot be killed by WBC, and thus definitely it will cause pneumonia. And when it will cause pneumonia, then the organism in which it has been infected will die. So when it was introduced in mice, the mice was dead. Let us see rough avirulent. What is the structure of this? This is without lack of capsule, so it is irregular in appearance. That is, it shows rough, so it is known as rough. It can be easily engulfed by WBC, so it can be killed, and so it will not cause pneumonia. So the mice will remain live. In the second case, Griffith did what he did. He has taken the virulent bacteria and heated up till 60 degrees Celsius, so the bacteria was killed. And when it was introduced inside the mice, there was no pneumonia and mice was live. Then he took the third case. In the third case, he has taken the smooth virulent heat-killed bacteria. He has mixed with the rough non-virulent bacteria and he made a mixture. This mixture was introduced or infected to the mice. The mice over here died. The mice should not die because both are dead. So why mice? Here the mice was dead, though there was a mixture which was having a heat killed, that is this one, and this was non-virulent. The question was in the mind of Griffith that why it happened. When blood test was done, in the blood test, the result was that S3 was seen. The question was arisen that how this S3 can be there when we have already taken S3 heat kill. So here it was believed by Griffith that maybe the S3, the heat killed bacteria, had some factor that was known as transforming principle that might have changed the R2, that is rough bacteria, into the S3 bacteria. This is because the synthesis of the smooth polysaccharide capsule might have been done by this factor. Later on, the three scientists, Avery, MacLeod and McCarthy, continue Griffith's experiment and they tried to prove the transformation principle. They took the S3 bacteria and they have ground it finally, and all the fractions of this bacteria were separated. Three fractions were there. One was DNA, second was protein, and third was carbohydrate. All the three components, they were added in the 3 Petri dish which contain the mixture or the R2, that is non-virulent bacteria. So here blue are the non-virulent bacteria. In this the components are added. The result was that only in the first case, the first Petri dish, there was a change of R2 into RS3 type, that is virulent type, while there was no change in the rough 2 bacteria. Here they remained as it is. So he concluded with this particular experiment that DNA can modify hereditary material from one organism to another. They again did one more experiment related to this. In this, they have taken 3 enzymes. Number 1, DNA hydrolyzing enzyme, and this was added in the first petri dish. In this one, when DNA hydrolyzing enzyme was added here, the DNA was inactive. Due to this particular R2 was not able to change into S3. In the second petri dish, they have added the second enzyme, protein hydrolyzing enzyme. And in the third petri dish, they have added RNA hydrolyzing enzyme. There was no effect. The R2 remains R2 only. There was no change. So they concluded from this particular enzyme experiment that it is the DNA who modifies or can change the hereditary material of one organism into another, and this was known as transformation or transformation principle. Now, we were not yet confirmed whether it is DNA or protein who can. Hershey and Chase, they did an experiment on bacteriophage. What is bacteriophage? So, bacteriophage is the virus which can infect the bacteria. So, this is the structure of a virus shown here. It is made up of hexagonal structure, is made up of protein coat which contain inside maybe double helix, it may be DNA, or single strand, then it is RNA. And they have a tail and the filament. These filaments are needed to attach on the bacteria. If virus is having the RNA, then you should know some of the examples of it. They can be tobacco mosaic virus, or it can be QB bacteriophage, or paramyxone, or hepatitis B virus. Now, Hershey and Chase have taken bacteriophage T2, which attacks on Escherichia coli, that is E. coli bacteria, which is seen in the colon of human intestine. Now, let us see here. This is the test tube. 2 test tubes are taken which contain some of the nutritive media. In this, they have added sulfur radioisotope 35. And in another test tube, they have taken nutritive media containing phosphorus 32. And now, this bacteriophage was allowed to infect E. coli in their nutritive medium. After few hours, it was seen that this bacteriophage has taken the S35 radioisotope, as well as this bacteriophage has taken phosphorus 32 radioisotope. We should know that S35 was taken by the outer protein coat because it is made up of amino acids cysteine and methionine, which needs sulfur. Over here, DNA has taken the phosphorus because DNA is made up of phosphorus. Now these 2 bacteriophage are ready with us with their own radioisotopes. Now they were allowed to infect E. coli bacteria. After the infection, this was the result seen. Now, this bacteria and this virus both are connected. So, blending was done to separate the protein coat with the bacteria. So, here the blending is done of both of them. Then it was allowed to settle down by centrifugation, due to which, as protein coat is lighter, will be on the top floating, and the bacteria cell will be it is settled down because it is little heavier. It was seen that inside the bacteria there was no radioisotope found in this case, while in this bacteria the radioisotope phosphorus was seen. So what is the conclusion? So here we can say no sulfur 35 isotope inside the bacteria means protein did not enter inside the bacteria, while here DNA entered the bacteria cell. So we can conclude here that it is DNA, not the protein, which is the hereditary material.